graduates.
Good morning. Today I'll be talking about the skin barrier and also immune dysregulation and atopic dermatitis. I forgot to say I have no disclosures, so pretend that that's the next slide. The outline will be just a brief background of atopic dermatitis. I'll discuss the skin barrier, some current research that's going on there. Then I'll move into the immune dysfunction, which includes some new therapies that may be coming along. And then I'll do a summary, answer any questions. So atopic dermatitis, as we all know, is just a very itchy, chronic disease state that we have a lot of patients suffering from. It's the most common skin disease of children. It occurs in about 1 out of 4, and in adults it affects about 10%. The onset is usually at less than 5 years of age in about 90% of the cases. It involves the face, the extensor surfaces, and extremities in children younger than 2 years old. And as you get older, it tends to involve the flexural areas of teenagers and adults. Greater than 50% of individuals affected with atopic dermatitis develop asthma and other allergies. They call that the atopic march. And it's a Th2 predominant pathway that causes inflammation of the skin. So the major clinical features, which are also the major diagnostic criteria, are itching, chronic or relapsing course, and a personal or family history. Other features are early onset, dry skin, hand and or foot dermatitis, cheilitis, which is that cracking of the lip corners, nipple eczema, and also immediate hypersensitivity skin responses. That goes along with that. Excuse me, atopic march. These are just some pictures. I'm sure everyone has seen eczema in their practice. But this just demonstrates on adults, you see it on the wrists and the popliteal fossa, and on children there you see it cheeks and neck. Known triggers are food. About 25 to 30% of those with moderate to severe disease. The most common foods are milk, egg, soy, wheat, fish, peanuts, and tree nuts. Aeroallergens are also triggers, in particular dust mite, animal dander, weeds, and molds. Those are kind of the bigger ones, unless you're rolling around with rats. Autoantigens, so IgE antibodies against yourself, can also trigger this, and then microbial agents and toxins. And one big example is staph aureus. This is just a pictorial way to treat eczema. So basically, you start with something like a cream or a lotion. If that doesn't help, then you move up to topical steroids. You can increase the strength and currently, there's only basically 3 systemic therapies that are approved depending on which country you live in, and that includes steroids, so prednisone, oral prednisone, cyclosporin, and then PUVA treatments, which is just basically light therapy. So currently, we're still trying to figure out what exactly causes atopic dermatitis. There's the inside-out and outside-in hypotheses. And so basically what they've come up with is impairment of the skin barrier, which leads to an increased responsiveness to allergens and an impaired innate immune response to pathogens. So you could read it that way, or you could read it in the reverse way for the inside-out hypothesis if you prefer. But basically what it comes down to, we're still trying to figure it out. This is just a picture of normal skin. And the reason I have this is it'll help when I talk about the skin barrier. So there's stratum corneum, that top layer of keratinocytes, and then there's lucidum and the granulosum, and then the stratum spinosum, basale, and then dermis down here. So stratum corneum and the stratum granulosum are the 2 areas I'll be focusing on. This is a cartoon of the skin barrier showing just the skin barrier itself, and then all of the immune actions that occur underneath it. So to start, I'm just going to start right here, which is the stratum corneum, and then we'll get into this stuff a little bit later. So the stratum corneum is composed of keratinocytes. These produce lipids and proteins to minimize water loss and also protect us from allergies or microbes. There's dephosphorylation and cleavage of Filaggrin by serine proteases, and that produces the filaggrin, which is important in those activities that I mentioned above. And other important proteins are Filaggrin and Filaggrin. I apologize if I'm pronouncing these wrong. Also, the skin barrier contains tight junctions in the stratum granulosum, and this provides a second physical barrier. Defects in this barrier contribute to skin inflammation as well, atopic dermatitis. So now just talk a little bit more about Filaggrin, which I'm sure everybody has heard of. It aggregates the keratin cytoskeleton for that stratum granulosum. It's proteolyzed into byproducts, which help to make natural moisturizing factor, NFM. And the breakdown products also play a role in acidifying the stratum cornea. So the increased pH activates serum proteases and leads to a barrier breakdown. So you want to keep that pH lower when we talk about skin. This is just a table showing the factors that can reduce levels of epidermal profilaggrin, filaggrin, or both. What I wanted to point out here is it's not just genetic defect or even the immune system, but actual environmental exposures, topical therapies, and microorganisms can also cause a decrease in filaggrin. So even if someone doesn't have the FLG mutation that we've heard about, they can still have reduced levels of filaggrin, which can potentiate atopic dermatitis. And then that brings me to FLG, or filaggrin mutations. So about 10% of Northern Europeans are heterozygous carriers of this mutation, and these are lots of function mutations. And about 3 to 6% of certain Asian ethnicities also have FLG mutations. They've found that the copy number variations correlates with levels of uricanic acid, and that increases your risk of necrosis in atopic dermatitis. So as those copy numbers decrease, then that uricanic acid decreases and you have that risk of dry skin. Also, they found epigenetic regulatory defects. They found that depending on how the DNA is methylated and adjacent site to FLG, that can also cause issues with filaggrin. Those are some new things with that protein. Deficiency of filaggrin results in a thickened stratum corneum, that layer that I mentioned at the beginning. The granular cells are reduced, and you also get decreased production of tight junction proteins, in particular gluten. You also get impaired lipid secretion and an increase in skin pH. Because as the pH increases, you get serine proteases activated, kallikreins, and then that just potentiates all of its effects. So it's understandable why filaggrin was focused on for so long, because of all of the duties it has in the skin.
Are there any products that have uricanic acid in them to use topically?
I am not sure about that, and I did not see any research talking about such. Most research right now is looking at biologic, so I don't know as much about topic. I do know CeraVe and some of those do have ceramides in them. I'm not sure. This is just a picture. So you can see as filaggrin decreases, whether it's inherited or due to environment, you get all kinds of bad results. Most of which I think I talked about. And a lot of these end up leading to Th2 inflammation and kind of that outside-in hypothesis. Something affects the skin barrier, and then that in turn leads to immune dysregulation. Also, other key observations that place filaggrin at the center of atopic dermatitis, etiopathogenesis, is that about up to 50% of Irish children with moderate to severe atopic dermatitis carry at least one mutation in Allergy. Even a slight reduction in the copy numbers from 12 to 10 increases the risk of atopic dermatitis. Mutations increase the risk of severe atopic dermatitis by all criteria such as early onset, persistence, infections, hospital visits, and use of medications. Filaggrin is downregulated in both lesional and non-lesional skin of patients with atopic dermatitis. It's also downregulated in lesional skin of darker skinned African subjects despite the lack of mutations. And the thought is that this may have something to do with vitamin D processing. And then coal tar is a useful therapy for atopic dermatitis, can restore filaggrin expression in the FLG haplo-sufficient karyotypes as well. However, I have not seen coal tar sold necessarily over the counter, so as far as topical agents, that's probably not a fun patients to use. So now we'll be talking about the stratum granulosum, and in particular the tight junctions that are found there. So these are found on opposing membranes of the stratum granulosum keratinocytes, which is directly below that stratum corneum. So again, kind of that second layer of defense. They're composed of adhesive proteins that control the passage of fluids and solutes through the paracellular pathway. One protein in particular that's been looked at is Claudin 1, and there's an inverse correlation between Claudin 1 and serum IgE. The defective structure may attribute to immune dysfunction, and that's kind of what the studies are looking at now in regards to those proteins. Also, interestingly enough, Langerhans cells can gain access to foreign antigens by sending dendrites out through the epidermal tight junctions, and so they're more likely to sample surface antigens and allergens if the tight junctions are compromised. So that kind of plays a role when we think of all the reports or studies that well, if someone's got peanut on their skin and they've got atopic dermatitis, can that lead to sensitization, etc. Claudins are basically 4 transmembrane spanning proteins that determine tight junction resistance and permeability. There's more than 24 members to this family, and some enhance the barrier and some disrupt the barrier. So it's a little tricky. 1 and 4 are good Claudins, 2 and 6 are bad Claudins. In regards to atopic derm. And they've done studies with Claudin 1 knockout mice, and they died within 24 hours of birth due to dehydration because their skin barrier was so broken down. With these mice, there were no abnormalities in the stratum corneum. So that's a kind of a key point there. It's not something else going on that they found in particular. It's tight junctions. So expression is reduced in non-lesional skin of patients with atopic derm compared with non-atopic and psoriasis patients. Levels are inversely correlated with Th2 biomarkers, and the analysis of Claudin 1 haplotype tagging SNPs in 2 North American populations an association of atopic derm. This is some pictures showing a study that did biopsies of individuals and then stained them for Claudin 1, and then also did some microarrays looking at genes present in these skin biopsies. So A and C are individuals with atopic dermatitis. B and D are individuals without. And the red staining here and the immunofluorescence are Claudin 1. So you can see obviously in atopic dermatitis patients, there's a decrease in this protein as compared to individuals with normal skin. Also, they found that as Claudin 1 expression decreased in Claudin 1 level, it was higher at a higher total IgE, higher level Claudin 1 had a higher total IgE non-atopic. It's the green. It's confused on my colors here. And then atopic individuals, which is the orange here, had higher IgE, lower Claudin 1 expression. The only problem with this is that they took healthy skin biopsies and exposed them to Th2 cytokines, and that actually caused an increase in Claudin 1 expression. So they're not exactly sure if this is cause or effect right now.
What's the PS on those slides?
Psoriasis. Yeah. Basically, they're just trying to show that atopic dermatitis and psoriasis are different pathways. And that brings me to immune dysregulation, which is kind of the other big player in atopic dermatitis. The Claudin 1 and Filaggrin were the big proteins that they're looking at now in regards to skin barrier, and then a lot of research is being done immune dysregulation. So as you can see, we've got our allergens, we've got our microbes, we've got the scratch and itch, and then all of that leads to barrier breakdown, re-traumatize it, and then this immune dysregulation. So basically, they've done a lot of different studies looking at large amounts of skin biopsies in atopic dermatitis patients, and they found that it's a Th2-mediated inflammatory response in general, and then they just kind of went from there as far as trying to figure out exactly whether to target and what to play in a bigger role, or that, etc. One of the things they found was toll-like receptors. So these are expressed by keratinocytes and also antigen-presenting cells. They function as pattern recognition receptors. They're stimulated by microbes or tissue injury to release antimicrobial peptides, cytokines, and chemokines, and they found that a deficient production of antimicrobial peptides is present in atopic dermatitis patients. These TLRs also enhance the strength of tight junctions. They may also play a role in that actual physical barrier along with the immune issues. So I just wanted to highlight that here. These are the Langerhans cells and then dendritic cells, and that's what I'll be talking about next. And take note, those Langerhans cells are able to project pretty far up the skin there. So if your tight junctions are not working appropriately, you can see how much they can pick up in regards to allergies or so Langerhans cells express that Fc epsilon R1 receptor that binds to IgE, and also there's inflammatory dendritic epidermal cell, or IDEX. So Fc epsilon R1 activated IDEX stimulate naive T lymphocytes to become interferon gamma producing T lymphocytes, and this in combination with IL-12 and IL-8 leads to a switch from Th1 to Th2. IL-23 is a cytokine that's produced by dendritic cells. It induces the production of IL-17, and currently they're doing studies with ustekinumab, which was actually produced for psoriasis because of that IL-17 pathway, and they found that the target shared by IL-12 and IL-23. So going back to that IL-12 here, switching it from Th1 to Th2, they thought if they targeted this potentially they could help mediate atopic dermatitis. There was one study that showed 4 males all had improvement on ustekinumab, and another study that showed one 16-year-old female had improvement, but otherwise it's relatively variable at this time. It's not yet. So next I'll be talking about IgE in regards to immune dysregulation and atopic dermatitis.
While you have that slide up, I was curious, why does applying, I'm talking about the peanut patch that seems to be therapeutic, so it has favorable data. Do they know what the mechanism of action would be to apply peanut to the skin and produce downregulation of peanut allergy?
I don't know the exact mechanism. I assume it would be similar to other desensitization where your body gets used to it over time, but I don't know the pathway that it's...
Because everything you're about to tell us is stimulatory.
Yes.
Applying an antigen to the skin is picked up by dendritic cells and it's pro-inflammatory to an allergic reaction, and it seems counterintuitive then to put peanut on the skin and produce just the opposite result.
Is it the dosage?
Yeah.
Frank, do you know?
Well, I mean, I think it's the dosage phenomenon
Yeah.
where it's micrograms, where you get sort of like with skip, you get sort of a paradoxical, down-regulatory effect.
And IL-10 is a regulatory cytokine that's involved with atopic dermatitis.
I was just going to say, it's very confusing sometimes what you were showing about the IL-17 pathway. It's, I think a lot of people think of that as being kind of pro-inflammatory, but there's several papers recently that show if you exogenously give IL-17 in asthma models, it's anti-inflammatory.
Yeah.
So
And that's where a lot of the studies are facing issues, because what they thought would always be inflammatory or found here is not necessarily what they're finding. So just like that study with Claudin 1, it's really unsure Is it Claudin 1 defect that's causing immune dysregulation? Is there immune dysregulation that causes Claudin 1? Are they not even related? So they're finding a lot of information, but then brings up more questions of how these cytokines actually react. Basically, we don't. To answer your question. So about 80% of atopic dermatitis patients do have an elevated IgE. Rituximab was tried, thinking that if they target the B cell that produces the IgE, potentially that would help. There was a decrease in IL-5 and IL-13, but no change in the overall IgE level. And no significant standard improvement or reliable improvement in atopic. Omalizumab treatment effects have been inconsistent without a significant benefit. The treatment does reduce IgE and itching, but did not reduce skin disease severity. One study, however, did look at this, and they divided the FLG mutant atopic dermatitis patients from those without, and those lacking mutations actually did have improvement with omalizumab treatment. So it may be more of a phenotyping issue than omalizumab not.
Erin, I think another issue just looking at Zoller a few years ago is the dosing was often, based on the asthma grid. And it did show that if the sort of total IgE was less than twice what would have worked on the asthma grid, you did get a positive effect. In other words, you weren't blocking probably enough of the IgE.
And so I think you're correct, and that's what's leading to these results that aren't consistent.
Right.
But I did look at the study that split out the FLG mutation carriers from those without, because that was the one that was relatively consistent in regards to those without did see improvement. And so as you can see, this is just a breakdown of the patients involved. The mutation carriers are over here, the non-responders, basically. None of them responded. And those without mutations did respond. That was a bonus. They did SCORAD instead of EASI, and this was their score at the start of therapy, and then this was at visit 14. And so basically it was 20 adults. They did 14 cycles of 150 milligrams of omalizumab in 2-week intervals, and if they didn't improve their SCORAD by greater than 25%, then they increased their dose of omalizumab to 300 milligrams. And so as you can see, in those who did not have the FLG mutation, they had significant improvement of their SCORAD. And then this is, right here, a lot of these studies looked at TARP, and so I had to figure out what exactly they meant by that. And basically, it's a thymus and activation regulated chemokine. It's chronically activating the thymus, but only transiently in stimulated peripheral blood mononuclear cells. It binds and induces chemotaxis in T cells by interacting with the chemokine receptor CCR4. It's also known by the name CCL17. So that makes a difference. Remembering this information, but CCR4 is expressed on Th2 T lymphocytes. It's upregulated by T cell receptor activation. Some reports suggest a role for this receptor also in trafficking dendritic cells. And then the chemokines that signal through this receptor are CCL3, CCL5, CCL17, which is TARC, and CCL2. And so that's why they're looking at it in atopic dermatitis individuals. It's basically to try to figure out if that Th2 pathway is upregulated. So this is what they found in omalizumab patient, that study I was referring to. IgE did significantly decrease in all of them, whether they were FLG mutant or not. The non-responders are in the lighter color, and those would be all the FLG mutants. And then the dark are the responders. They also looked at TARC to see what the inflammatory response was doing, and they found that that also decreased in both study groups, but again, that did not improve those with FLG mutants. So back to my favorite slide, immune dysregulation. Next I'll be talking about the actual T cells involved. So we went from kind of the general skin cells to B cells to now T cells, so it's a Th2 and Th22, and you can see all the effects that these cells have in regards to immune system and skin. So basically, epidermal keratinocytes express thymic stromal lymphopoietin, or TSLP. They also express IL-25 and IL-3. These are cytokines which enhance dendritic cell-driven Th2 cell differentiation, and they also increase in response to mechanical injury, allergen exposure, and microbial infection. The Th2 cytokines in particular are IL-4, 5, 13, and 31, and then the Th2 cytokine is IL. All of these cytokines are found to be elevated in lesional skin biopsies of atopic dermatitis, and mouse models will develop atopic dermatitis skin signs and symptoms if you increase the levels of these cytokines or knock out the regulatory counterpart. So in particular, IL-4, IL-13, 22, and 31 contribute to the decreased barrier proteins, in particular Filaggrin, and also decreased antimicrobial peptide expression. So this allows enhanced penetration of allergens and pathogens into the skin because that barrier is disrupted, and you also get increased susceptibility to skin infections, particularly staph aureus. IL-31 also causes pruritus. IL-5 leads to eosinophil differentiation, activation, and proliferation in the skin, and then IL-10 plays an immune modulating role, but I couldn't find as much information describing exactly what that indicates. I just know it affects the T regulatory cells in this pathway. So next I'll be talking about the cytokines in particular. First off will be IL-4 and IL-13, and I highlighted here how many places these cytokines are found and all of the actions that they're doing. So they're decreasing our protective proteins, they're activating our inflammatory cells, and they're perpetuating basically this entire inflammatory pathway. Also, they're finding those innate lymphoid cells in skin as well. I didn't find a lot of studies focusing on that, but they're present. So those cells that Sydney had talked about earlier this year, kind of that new population, those may also be something. Sure. So basically, IL-4R alpha, or IL-4 receptor alpha, is a common receptor for IL-4 and IL-13. It induces the STAT6 JAK1 signal cascade, and currently they're trying to, and as you can see, those are all the effects that these cytokines have in the skin, and so they're trying to target that fully human monoclonal antibody called dupilumab, and that basically targets that receptor. They've found in several studies that weekly treatments lead to reduced severity and also a decrease in skin infections, and the molecular changes in the skin actually paralleled the improvement in clinical scores. So basically they took individuals who were not adequately controlled with medications, had moderate to severe atopic dermatitis. They used dupilumab as a monotherapy in 2 4-week trials and also 1 12-week trial. The endpoints included the eczema area and severity index score, the investigator's global assessment score, the pruritus, and also safety assessments, and then also serum biomarker levels in the disease transcriptome. This is just another picture showing basically they're trying to block this with dupilumab. So all of the functions of these two cytokines, they're trying to basically take that out, see if it improves. Yes?
Has anybody tried applying these topically with something like DMSO that would drive it through the skin instead of giving them intravenous doses?
Not that I know. So, and this one is, so the study that I found with dupilumab was in New England Journal of Medicine. Not only was it kind of a phase 1 trial, but they're also looking at safety. So I don't know if I didn't see any report of other topical therapies or anything else in it, and maybe something they can provide. So basically, these were the results of that study. So the top here is in the 4-week studies, and the reason there's 2 of them is one was in the United States and one was international. That reported in, and they did different dosing of dupilumab, starting at 75 and going up to 300 milligrams as compared to placebo. And as you can see, so this is their eczema area severity index, and they wanted it to improve by 50% or greater. That's why 50 is there. And this is the study day and the percent of patients. So the longer they were on it, the more they improved, and it was dose dependent as far as how much improvement. And then that also was pretty much the same as in regards to their itching scale. So again, their average weekly pruritus scale decreased in those who are on dupilumab as compared to placebo, and the higher dose more decreased itch. Then they looked at a 12-week study and basically confirmed the same thing. It's continued to have improvement over the 12 weeks as compared to just the 4. So you can see here, kind of the top line is about 70%, while if you continue it for 12 weeks, you can get up to 80 to 90% improvement while on 100 milligram dose. Same over here with pruritus.
And that's an IM injection monthly?
So they did it weekly, it's every 2 weeks.
Every 2 weeks an injection.
Yeah.
Well, that slide looks like there was not a dose response. That looks like the lower dose, the 75 did better than the 300.
Look at the B.
B. Oh, I see what you're saying. Right here where it crosses over.
No, it's not
Well, but
something
all the first 2 weeks or so.
It's not significantly different.
It's interesting.
So in the air.
Oh, I see what you're saying. 75.
Yeah.
Gotcha. Yeah. So, it may be that in a lot of these, with even omalizumab, people would have improvement of their itching, but not necessarily improvement of their skin severity. So it might be one of those effects that we see with overdose.
While you bring up itching, I remember when John Milner used to join us. He was a dermatologist who passed away, a lovely man. He always used to say that pruritus was not caused by histamine in this disease, and antihistamines didn't help. He never really told us what did help, though.
They found that some of the cytokines actually stimulate the neurons, and that causes a lot of the itch as well. And that obviously doesn't have any histamine.
So from a pragmatic sense, what's good for itching in this condition?
Anything that works.
Interesting. Gabapentin treats itch.
So again, they looked at my favorite TARC or, in these individuals as well. And you can see, so they measured it at baseline and then followed it over the course of time. And, again, with the 75 milligram dupilumab, they've had some odd measurements here. But with the other doses, looks like TARC decreased from baseline. Not sure what's going on with 75 milligrams. So we just pretend that we just don't ever use that dose. Right. So the next one is, again, this is a 12-week showing the same. So over time, and that the 12-week was all 300 milligrams. There was no dosing change outlet. But they did have improvement of measurement of TARC. As a side study, branching off of this one, they also had several patients sign up to have skin biopsies performed so that they can analyze those as well. And what they found is that dupilumab improves the molecular signature of the skin of patients with moderate to severe atopic dermatitis. So basically they had 18 patients total, and these were all from the 4-week studies that they did, as compared to those who did not have the drug. And so you can see here, basically they just had a decrease in that eczema area score that they're talking about, and that correlated with specific genes. So upregulated and downregulated. They've done multiple studies looking at skin biopsies to try to determine the difference between atopic dermatitis individuals and normal skin, and they found certain basically genes that are upregulated or downregulated in atopic dermatitis as compared to normal skin. And so they were looking at the changes in those genes over time in people who had dupilumab. And basically in a nutshell is that they showed improvement in the atopic dermatitis lesional transcriptome that they were measuring over time with the dupilumab. And again, it looks like the 150 and 300 milligram were the doses that showed improvement. The 75 didn't include. Basically, just a molecular way to back up those symptom scores that they did. This is a little bit easier to read, in my opinion. So basically, they looked at certain things. So this is placebo/normal skin in an individual on dupilumab. This is at 150, and so red means that it was upregulated, green means it was downregulated. And the size of the font is basically how much or how little. So as you can see over here at the 300 milligram level, all of those cytokines that I've been rambling on about were downregulated. Dupilumab, which is a good sign, meaning that the inflammation, that immune dysregulation, is getting downregulated use of dupilumab. So hopefully something that we can look for as a treatment in the future.
In psoriasis, they've noted some of the microRNAs are differentially expressed. Have they noted any problems in microRNA expression in atopic dermatitis?
I know they've done microarrays, and I don't know exactly what you're asking for.
Just the microRNAs that control the gene expression of the, maybe one microRNA will control 50 to 100 of these genes.
Oh,
so there could be just one that's dysregulated. You could target that as a therapy.
I did not see a lot of that reported. I'm sure they've done it. But not something in the newer therapeutic set that I saw. So that brings me to cytokines IL-5 and IL-1. Mepolizumab has been shown in studies to reduce eosinophils, but did not significantly reduce skin disease severity. Again, it did reduce itching, but not the actual skin disease itself. Levels of IL-31 correlate with disease severity, and so trials using monoclonal antibodies targeting IL-31 are currently TSLP. This induces Th2 cytokine expression in serum IgE. It triggers IL-5 induction and eosinophil recruitment. A mouse model of TSLP-induced atopic dermatitis showed that it's T-cell dependent, but not IL-4 or IL-13 dependent. So it may be that dupilumab doesn't work in some individuals. TSLP also triggers the itch response via sensory neuron, and AMG 157, MK9 226 are antibodies targeting TSLP that are currently in investigation. This brings me to the Th22 pathway, cytokine IL-22. This is a T-cell, Th22 is a T-cell subset in atopic dermatitis patients that produces IL-22, but not IL-17 or interferon gamma. And it belongs to the IL-10 family of cytokines. So again, that comes to the debate of, well, IL-10 is a regulatory cytokine, why is IL-2, which is part of the family, not? And leads into that whole issue again. IL-22 induces STAT3 signal pathway, which causes epidermal thickening. It's present in chronic lesions, so maybe this is a marker of chronic atopic dermatitis versus acute, which could be that as that IL-10 family tries to regulate things, maybe IL-2 gets upregulated instead of fixing the problem, just causing these chronic lesions. And currently, ILB-094, the monoclonal antibody targeting IL-22 that they're looking into in regard to treatment. These are a couple tables showing all of the different studies in atopic dermatitis that are looking at using biologic therapies. As you can see, there's quite a lot. And so hopefully we'll be getting some good data and maybe some FDA approvals in the future. And again, it's just mostly targeting currently the immune dysregulation and not as much focusing on that skin. Just another slide showing more. These are all biologic that have been approved for other diseases, and now they're trying them in atopic derm to see if it would produce any type of significant result. So in summary, there's inside-out versus the outside-in hypothesis. The inside-out hypothesis is that immunologic abnormalities such as FG mutation predisposes the individual to IgE-mediated sensitization leading to atopic derm. And then there's also the outside-in hypothesis that the barrier abnormality, whether it's from heat or scratching, infection, predisposes an individual to atopic derm. After looking through all of these different studies and mechanisms, my personal belief is it's a combination of the two. You can't put it on one or the other. Atopic dermatitis is both structural and immunologic. In the future, it looks like the next big thing will be patient phenotyping based on skin biopsies, continued discovery of genes, and hopefully novel monoclonal antibody therapy or some new topic. So I see a lot focused on that. Any questions?
How about systemic methotrexate? I've seen somebody from children's whose kids have been put on.
Yep. So there are a lot of different immune suppression therapies that have been used, but they're not currently approved for atopic dermatitis.
Have you seen any studies with the methotrexate, or is
Cyclosporin was the main one that has been studied. I did not see a lot of methotrexate. There were a couple others. They tried TNF inhibitors, IL-6, but they had issues with infections or problems when you suppress the immune system. There were a couple other monoclonal antibodies on the market that have been removed because of PML or other issues, and those did produce some results in atopic derm, but again, not enough to overcome the side. So ideally, we'd like to have something that helps and doesn't harm.
Yeah. How does light therapy work?
I'm not sure.
Does it work?
According to Mayo, where I train, that's like their mainstay of treatment. They have
I think it's because of vitamin D.
Vitamin D.
Yeah, I think it's vitamin D. I think that's the mechanism is that they think that light therapy works by increasing your vitamin D levels.
Yeah, that T cell trafficking with the therapy, like they use that for the graft versus host disease as well. T cell.
All right, I want to make a comment.
Modulates T cell trafficking in the skin.
I think that's the thought.
So I want to make a comment. I'm just curious, like, you know how when they always say that like a third of the kids have moderate to severe eczema, have coexisting food allergies, and then they start listing off all the foods?
Yeah.
I mean, I personally do not believe that peanut or nut allergy is a trigger for eczema, because I think that kids who truly have peanut and tree nut allergy, they have allergic reactions. I mean, I think you find sensitized skin, but I don't think peanut allergy is a driver of eczema. I think it's egg,
Right.
milk, maybe wheat, maybe soy. But that's it. I mean, I always think it's egg, because it's always, yeah. So anyway, I don't know if anyone else has found that, but it kind of always drives me crazy when people say, you know, there's this peanut allergy, and I'm like, I don't think they have peanut allergy. They might be sensitized, but I don't think, most of them are not even eating peanut.
It's hard to know too if it's the actual peanut or just the irritation to the skin.
That's the other thing, is I think that you test these babies, and like they always have positive tests, but I don't know what's real.
Yeah.
Right?
Right.
I think the same is true. You had a slide earlier of what aero allergens are responsible, and I didn't agree with everything on that slide. I think dust mite is probably true. You had pollens up there. I was going to ask you, what data anybody has that pollen or mold, especially mold, I don't think has anything to do with anything. But do you know what data goes into the statement about aero allergens?
I do not. I just know that this is what was present in our board review. And so you probably have to know.
You know, Mary?
Well, I think, I mean, I have kids that, like, let's say that all their skin tests are negative except for the cat. You know what I mean? And so that's like, it's a little baby that has eczema, and I'll test for dust mite and cat plus whatever foods I'm going to do. And, it might be the only thing that comes out. So I do think that's true. And then I think older kids, I have kids that flare with their eczema when the pollen count is high, but not babies. But older kids, like a 10-year-old who outside playing, all of a sudden, next thing you know, and I don't know if it's because he's rolling in the grass with the baseball field or whatever, but I think for older people, older kids, but I don't think there's a lot of studies.
And you do autologous skin testing to see if these people have autoantigens against themselves, or is that not helpful?
I've never seen it done. I did not see any reports of that, but you could.
And the second question is, they used to treat mycosis fungoides with electron beam radiation, which basically takes out most of the surface layer of cells. Has anybody looked at that and see if you can wipe out the T cells that are intraepithelial instead of just on the surface?
I don't know. Study. Good question.
Actually, while you bring it up, the most recalcitrant patient I've ever had with what was billed as urticaria. This woman failed everything. Fusions of Xolair, plasmapheresis, but it looked clinically like urticaria. Ultimately was diagnosed with mycosis fungoides. Initial biopsy was non-diagnostic. So this woman, I finally had to send her on to a hematologist, but seen anybody with that confusion of urticaria, mycosis fungoides, Sézary syndrome.
It's just they seem to be really responsive to this electron beam radiation, but it makes sense like everything's T cells, so if you can find a way to kill the T cells in the skin, it might be helpful.
Just have to. I guess one of the problems then be, are you actually like what is that going to do for immune suppression?
And like the animal models, have they tried manipulating the microbiome to try to get rid of the staph, just to as an alternative approach? Or any human studies with regard to that?
I believe there were mouse studies because with the with some of the other triggers as far as the microbial agents and toxins, they would try those on mice to see, and they found like different toxins in medications, etc. But nothing that I saw recently. This isn't like just the last couple of years, so I don't out there microbiome. I didn't see a study directly addressing that.
Can Filaggrin be applied topically to try and repair this defect that people have?
Not that I found.
Because the one cream I've seen the dermatologists all like is CeraVe.
Yep.
And in 25 years, they've never agreed on anything else. So there's obviously something about that.
Yeah, it contains the ceramide.
Ceramide as well as an emollient. But I just wondered if Filaggrin is something that could be solubilized and applied topically to the skin as well.
Yeah, I don't know.
Lots of weird questions. And Marion, in terms of your question, I had a lady that was terrible eczema. She wasn't a kid, she was an adult. But when we tested her, she was just allergic to peanuts and chocolate. And her comfort food was basically peanut M&Ms. So every time